We're in section 6.2 and we're still talking about microbial growth. And this learning outcome is one of my favorite topics, and it seems a little challenging, but hang in there because there's actually some really interesting, relevant things about it. And it helps you kind of understand some things about oxygen that you never knew. So it says, explain how microorganisms deal with toxic oxygen products and the role of superoxide dismutase and catalase. So here we say, wait, toxic oxygen? We Oxygen is like a life-giving molecule, O2. We all need that to survive. But it turns out that the planet Earth evolved without oxygen. And so the first organisms of any kind, didn't have oxygen. They had to metabolize without it. And that's how we got that anaerobic respiration. There's a lot of organisms that have all sorts of crazy metabolisms where they don't use oxygen. And as evolution continued on the planet and plants or photosynthetic microorganisms evolved, then they started generating oxygen. Well, oxygen turned out that it was toxic to the existing life on the planet. So what had to happen is the organisms that were on Earth before aerobic organisms had to find ways to neutralize toxic oxygen. And we actually have those mechanisms even in our own bodies today. So oxygen is a toxic molecule, but our bodies and other life on this planet that lives in the presence of oxygen has developed ways to neutralize these toxic oxygen products. So let's look at this. What happens? Let me see. Let me get back. Okay, so without getting into too much detail about chemistry, what happens in the presence of oxygen is there is this stuff called free radicals or oxygen radicals that form naturally in environments that contain oxygen. So anyway, I won't go into how, but they do. And these things, these O2s with the negative sign, if you remember anything from your chemistry, the negative sign means it's an unpaired electron. And electrons, what they want to do is they want to stick to other electrons. They want to get involved with other electrons and create bonds. And so when you have these free radicals, they actually interact with other molecules, other organic molecules, and they disrupt them. They damage DNA, they damage proteins. And so you have to neutralize these oxygen-free radicals. Now, you have heard about these, whether you realize it or not, because you've heard that you're supposed to eat a lot of colorful vegetables and fruits. And you hear these, you want to eat things that have antioxidants in them. You've probably heard that blueberries are great to eat because they're one of our best antioxidants. Guess what antioxidants do? They neutralize these free radicals that naturally form. So all those colored fruits and vegetables, the reason why we're supposed to eat them is to prevent these oxygen-free radicals from damaging molecules in our cells and contributing to decline in our cells. So you've heard of them, whether you realize it or not. So what happens is these free radicals, and they interact with hydrogen. You don't even have to know that. But there's an enzyme that takes these and neutralizes them and turns them into, anybody know what H2O2 is? Hydrogen peroxide plus oxygen. So there's this enzyme called superoxide dismutase, SOD. These are actually called, I keep calling them free radicals, but you can also call them superoxide radicals. So these superoxide radicals, the O2s with the extra electron, by the action of this enzyme, it's got an ACE, there's that, is converted into hydrogen peroxide and oxygen. And we all know, oh, we've got hydrogen peroxide in our medicine cabinet. That's not dangerous. Oxygen, oh, that's good stuff. We use that. But it turns out, as you're generating hydrogen peroxide in your cells, you don't want hydrogen peroxide in your cells because we use hydrogen peroxide to kill bacteria. And that's because it is, in certain situations, it can be toxic. It can also be toxic. It's just not quite as toxic as these free radicals. So every um, organism on the planet that uses oxygen or lives in the presence of oxygen has to be able to take free radicals and neutralize them. They typically do it by making hydrogen peroxide and oxygen. So now we need a second enzyme that takes that hydrogen peroxide that our cells are making and neutralizes that. So what catalase does is it takes hydrogen peroxide and it converts those. So this enzyme is what catalase, and it takes hydrogen peroxide with the catalase and it converts it into water and oxygen. Now we've got molecules that are safely able to function in our cells. So organisms have to deal with toxic oxygen using the enzymes superoxide dismutase, taking free radicals, making hydrogen peroxide and oxygen. And then catalase is going to take that hydrogen peroxide that our cells made and make water and oxygen. And by doing that, this will allow cells to be able to live in the presence of oxygen and even use it in cellular respiration. There actually is another enzyme in bacteria, not in humans necessarily doing the same thing, but in bacteria that takes hydrogen peroxide and makes water and oxygen. It takes hydrogen peroxide and makes water, but not oxygen. And we can use an enzyme assay. We can test for that enzyme in bacteria, and we can use it to help identify bacteria because we know which ones have catalase and which ones don't. Even bacteria that don't have catalase have another enzyme that neutralizes hydrogen peroxide. So here's an example of a test that detects the presence of catalase. And you're going to do an unknown, a virtual unknown this semester. And I think there's a catalase test in that. So anyway, what they did here is they took a slide and they put some bacteria in two little spots. And then they put a drop of hydrogen peroxide on it. In this one, what you see are bubbles. And so what they're saying is this bacteria actually makes catalase. If you put hydrogen peroxide plus the bacteria that have catalase, you're going to get water and oxygen. Oh, oxygen's a gas. So when you take a gas and a liquid, you get bubbles. So here's a catalase positive one, and here's a catalase negative. You have hydrogen peroxide plus your bacteria, but no bubbles. So this means this organism doesn't make catalase. It must make a different enzyme that neutralizes the hydrogen peroxide. I also like to kind of mention, you think about it, a lot of people are aware that if you get a cut and you put some hydrogen peroxide on it, that you get bubbles. But a lot of people don't know why that is. Some people will say, oh, you got a cut, you put peroxide on it, it's bubbling because there's bacteria in the cut. But it turns out that is false. That doesn't mean you have bacteria in the cut. What it means is you have catalase in your blood. And it turns out a lot of bacteria and all humans have SOD and catalase in their blood. So if you take human blood and you mix it with peroxide, you're going to get bubbles. If you have a human that has a cut and you put hydrogen peroxide in the cut and you do not get bubbles, that person would not be alive. You cannot live without catalase. It's essential for life. I think there's some conditions. I thought I read about something that you might have low catalase, and that is not good. It can lead to different disease conditions. It's not very common, um, but we need catalase to live normally. So if you have hydrogen peroxide, you have a cut, and you put hydrogen peroxide in it, and it doesn't bubble, it means your hydrogen peroxide is bad. It doesn't mean you don't have catalase. You have catalase. Um, Hydrogen peroxide is always in a brown bottle because even when it's exposed to light, it will naturally decompose water and oxygen. So over time, it takes a long time. It takes what? Yeah, it takes a while. I save it for a year or more and I still can use it for a positive catalase test. So, okay. So understanding that, let's look at categories. We have 5 categories of microorganisms grouped by the way they use oxygen, and I want you to recognize their growth pattern in thioglycolate medium. Okay, so I'm going to get to that thioglycolate in a minute. But when you look at these words, if you look at them carefully, they tell you a lot about the organisms. So when we're looking at organisms that are aerobes, aerobe means oxygen loving. These are organisms that live in the presence of oxygen and use oxygen. An aerobic organism is one that uses oxygen. Okay. All those organisms, including us that do cellular respiration, are aerobes. So these are really common. Anaerobes, an means not. So these are not aerobes. These are organisms that do not use oxygen. And if you're an obligate anaerobe, I probably should have just left anaerobe off there and just put obligate anaerobe. But anaerobe means doesn't use oxygen. If you're an obligate anaerobe, you have to be in an environment where there's no oxygen. An obligate anaerobe has to be in an environment where there's no oxygen. Why? Because they don't have SOD and catalase. If an obligate anaerobe finds itself in the presence of oxygen, it's going to die because of toxic oxygen. It doesn't have SOD or catalase. Aerobes have to have SOD and catalase to survive, to live in the presence of oxygen, just like us. So aerobes grow in the presence of oxygen. Aerobes use oxygen and therefore grow in the presence of oxygen. Anaerobes do not use oxygen. Obligate anaerobes have to be in the absence of oxygen to even be alive. So they have to be in an oxygen-free environment. Here's a word, you're going to see this a couple times this semester, facultative. Facultative in microbiology means you can, but you don't have to. Facultative, you can, but you don't have to. So this is saying you can be an anaerobe, you can survive in the absence of oxygen, but you don't have to. Facultative anaerobes are very common in the microbial world. These are organisms that can live in the presence of oxygen, or if they run out of oxygen, they can grow anaerobically. They can grow by cellular respiration or fermentation, and they can live without oxygen. Now, remember which way they grow faster. Microorganisms will grow faster using aerobic cellular respiration because oxygen, you make more ATP, right? So if there is oxygen, facultative anaerobes will use it. And then when they run out of it, they'll probably start fermenting. That's the most likely way. They'll probably just start fermenting whatever's around them for energy. So, and they'll grow more slowly. So think of something like E. coli. E. coli is a really common fecal bacteria. There's all sorts of common fecal bacteria. We just all happen to know about E. coli. So, where do you find E. coli? Well, go out in my yard. I've got deer poop all over there. There's E. coli out in my yard. When that E. coli is sitting out in the yard, There's all sorts of oxygen. So the bacteria can grow fast using aerobic respiration. It can grow aerobically. When the E. coli is inside of the, the animal it came out of, there's not very much oxygen in the intestines. So inside of the animal, it's going to grow anaerobically. It's probably going to grow using fermentation. So E. coli is a facultative anaerobe. It can grow with oxygen if it's available, but if it's not, it'll grow without oxygen. These last two, people get them kind of mixed, confused, but look at the words. The words tell you what they are. Anaerobe, mean it doesn't use oxygen. Aerotolerant. Aero is referring to oxygen. An oxygen tolerant anaerobe. Remember, an anaerobe is going to grow without oxygen, but an oxygen tolerant anaerobe, let's see, if you don't use oxygen, but you can grow in the presence of oxygen, you would be an aerotolerant anaerobe. And what makes you aerotolerant is you have SOD and catalase. You've got enzymes that will detoxify oxygen, so you can live where there's oxygen. You just don't have the enzymes to use it to grow. So these are anaerobes, but they can survive in the presence of oxygen. Obligate anaerobes, they don't use oxygen. And if you put them in the presence of oxygen, they're going to die because they do not have SOD and catalase. Okay, here we go. Micro aerophile. Aero, oxygen, phile, lover. Micro, small. Small oxygen lovers. So these are organisms that they'll use oxygen, but they only grow in environments where there's low oxygen. So that might be like a place like a swamp, that there might be a little bit of oxygen in the water in some places, but there's not a lot of oxygen. There's a low concentration of oxygen because the water's moving slowly and oxygen isn't moving around, isn't dissolving into it very well. So these are organisms that like a small amount of oxygen. And the reason why they can only use small amounts is because they don't have as much SOD and catalase as aerobes, facultative anaerobes, or aerotolerant anaerobes. They have low amounts of SOD and catalase. So oxygen is a little bit toxic. So they can only survive in low oxygen environments. So in lab, you're going to be talking about these, they're called fluid thioglycolate tubes or thioglycolate tubes or thioglycolate medium. What that means is, here's you have a test tube and it's got this medium in it. There's some bacteria food. And it's semi-solid. And so what happens is the oxygen, when you heat it up to sterilize it, the oxygen's driven off. But when you put it back on the, on the lab bench, and air gets exposed to air, the oxygen from the air will dissolve in the medium. But because the medium's really thick, it's hard for the oxygen to get all the way to the bottom. So what happens is you end up with this oxygen gradient. You have a lot of oxygen up here, a little bit less, a little bit less, a little bit less, a little bit less, until you get to the bottom where it's anaerobic. There's no oxygen. So in this thioglycolate medium, you have high oxygen at the top and moderate, and then no oxygen at the bottom. So we have an oxygen gradient. What this picture is showing you is where there's lots of dots, that's where you have lots of bacteria growing. So if I actually took like a needle, I dipped it in a needle into my bacteria, and then I stabbed it all the way down to the bottom. So I inoculated this whole thing with bacteria, but the bacteria only grew at the top. Which of these organisms would I have? So let's cross off anaerobe. We'll just use obligate anaerobe. This question is actually in your study guide. Okay, so it has 5 tubes. It has this exact same picture in there. And I have the answer in your study guide, number 2. So I'm going to help you with the first one because it's pretty easy. It's just to get you started. If you look at this, the bacteria only grow where there's high oxygen. Oh, so that must be the aerobes. Okay. The other one to notice is look at these 2 tubes. This one is showing you the bacteria are growing faster at the top. There's more concentration, and they're growing slower down here where there's little or no oxygen. In this one, they might grow a little bit at the top, but they're going to grow better at this slightly lower oxygen concentration. So I don't want to just give you the answers. I want to see if you can figure it out. Try to figure the question out before you look at the answers and match these 5. Cross off anaerobe. We'll just use obligate anaerobe. And see if you can match these 5 with these 5 thioglycolate tubes. It's in your lecture study guide. Oh, yeah, so which of these need SOD or catalase? There's a table I put in, I think I did. Oh, maybe I didn't. Oh, yes, I did. It's in your study guide too. There's a table asking you which need SOD or catalase. If you're going to grow in these high oxygen environments, you need to have SOD or catalase. So there's only one that doesn't have SOD or catalase. So see if you can kind of put all that together. I'm going to stop right here.